Morning, everybody. All right. So yeah, a few more people have shown up. Hello, everybody. Okay, so hopefully you had a chance to watch the short video on transcription that I did last Thursday. Does anybody have any questions about any of that stuff that they'd like to go over before we continue? No? Cool. Okay. So, good morning, Tanya. Let's get started on this. All right, so this is where we pretty much left off. Again, remember that eukaryotes are quite considerably more complex than prokaryotes in the control of the initiation of transcription. Not to say that prokaryotes don't have some tricks up their sleeves. They do some pretty funky stuff, which we'll get to in the last section of the course on control of gene expression. But in terms of the number of players, it's much more straightforward in bacteria than it is in eukaryotes. Okay, so once this pony is off at the races, or greyhound or whatever, essentially it just keeps going, right? There's nothing intrinsic to RNA polymerase that is able to stop itself. It's a pretty dumb machine, to be honest, right? And the same with DNA polymerase too. And so there has to be some way of stopping it, right? Because otherwise you risk essentially transcribing all of the genome, or all of the chromosome rather, from this point onwards, which would be, A, it would be a terrible waste of resources, right? Because it takes a lot of energy to do this. But also it would risk expressing genes which you do not want to be expressed, right? Which may be on the same chromosome but have very, very different roles to the one that you're expressing. And so there has to be a way of stopping this. And so really elongation, which is kind of what happens after initiation, is pretty uninteresting. You know, it just happens. As long as there are RNA nucleotides present, RNA polymerase will turn them into mRNA, basically, or whatever RNA that polymerase is doing. And so really nothing much happens of note until this is terminated, right? And so I want you to get kind of, there's three kind of real main mechanisms of how this works, right? And in bacteria, it's really tied to formation of RNA secondary structures, right? So this is some, and actually that's common to an awful lot of processes in gene expression in bacteria, right? These RNA secondary structures, which are just there we go. It's even there in my search list, right? So this is what I mean by RNA secondary structures, right? So RNA base pairing with itself, right? So as it is produced, and typically it's the result of inverted repeats, which then kind of fold up and stick to each other, right? Because they're complementary. But these hairpin loops, like example you can see just here, are very common in bacteria, right? And they have a very prominent role in the control of gene expression. Come on, let me get back. Oh, I'm in my, I was like, where the hell's Zoom gone? All right. So there's basically a means of two different ways of terminating transcription in bacteria, and one that we know of in eukaryotes, right? And so in bacteria, there's basically intrinsic and extrinsic termination. And so both of these rely on features that are encoded in the gene itself, because those features will be produced once that part of the gene has been transcribed by RNA polymerase. And these features, their main role is to slow down or stop RNA polymerase, right? That's kind of where they're at. There are different flavors to it, but that's the kind of the overriding principle behind this. There's just different ways of doing it, right? So there's intrinsic termination, which is purely controlled by those inbuilt sequences. And there's row-dependent termination, which relies on both that and an external protein known as Rho, R-H-O, not Rho, Rho. Anyway. Okay, so, and again, if anyone has questions, please do feel free to stop me and ask. Okay, so intrinsic termination in bacteria relies on what are called an inverted repeat, right? So you probably remember these from transposons, right? So transposons also have terminal inverted repeats, right, which are recognized by transposase to cut out the transposon. Same kind of principle. And so essentially what you have here, if you read the top line, you know, AAGCCTCC, you'll have exactly the same on the bottom strand going in the other direction. AAGGCTCC, right? And so that means that the top strand here has a sequence, this sequence here along the top strand, which is exactly the opposite of the sequence and in the opposite direction. So it's a mirror image, right? So when that part of the gene is transcribed, because obviously you don't get a hairpin loop in DNA because it's already base paired, it's already double-stranded. It can't do that. But RNA is single-stranded, so as soon as it's produced and both of these sequences are transcribed, now they're free to associate, and now you can see perfect base pairing all along what's called the stem of that hairpin loop. It's called a hairpin loop because it looks kind of like a hairpin, right? Bobby pin or whatever you would call them. And there's always a few bases here which basically allow that kind of hinging, right? So that our mRNA can kind of hinge and associate with itself, right? Because there's just needs a little bit of space to be able to fold back, right? So this is really important to both intrinsic and extrinsic or row-dependent termination, formation of these hairpin loops. And so essentially what the deal is that there's a kind of a string of A's in the DNA sequence, right? And for whatever reason, I'm not entirely sure why, but when RNA polymerase synthesizes a string of uracils or U's, it tends to pause or slow down. And essentially what that does, and this is again the same as what we will see in row-dependent termination, is it allows a structure to form which kicks it off. And that structure is this hairpin loop. And so essentially the RNA polymerase is kind of coming along this way, right? As soon as it makes this string of U's, right now it's essentially paused kind of over this part of the transcript. And this hairpin loop actually forms inside the RNA polymerase. And it literally acts as a lever to just pop the RNA polymerase off of the DNA. Because it physically kind of forces its way into the channel. Where we got it? It's up here somewhere. Here we go. Forces its way into the channel and kind of forces the RNA polymerase to dissociate from the DNA. And so if you can imagine here, right, there's just enough space for this RNA transcript to be synthesized and then emerge kind of from the RNA polymerase. Well, in intrinsic termination, what happens is that there's a series of U's here, and then this part forms a hairpin loop and literally levers the RNA polymerase off of the DNA substrate. Because there's not enough physical space for that all to occur in the same spot. Does that make sense? No? Okay, let's see if I can draw this up.
Yes, because can you start over when it's like the mirror image? I kind of like, I don't get it. I really don't.
Oh, okay, sure. Sometimes this is something that's a little easier to, I'm not going to use the exact same sequence just because I'm going to be getting rid of this for a second. All right, I'm going to put the light on so there's a bit more. May just have 3 bright spots on the whiteboard instead. Wow. I wish they made matte whiteboards. Okay, so essentially what you have is, where did my blue pen go? Oh, here it is. Oh, let's see if we can have some more colors. More colors. All right, so essentially what you have is, here's your DNA, like so. And there's I'm not going to draw the other strand because it would just take up a lot of space. Essentially, you have, what would it be, GC, A, and then that would be T, G, C, and then we're going to have a bunch of A's, right? Just kind of making up as I go. And so, how about that? I got black in here as well. Forgot about that. So these little sequences here are the complete opposite of each other, right? So A complementary to T, G complementary to C, hang on, no, C complementary to G there, and G is complementary to C, right? So these, if they are free to do so, which they will be in the mRNA sequence, will want to stick to each other, right? Because A is, this is going to get copied into uracil. Uracil is going to want to base pair with A, right? So essentially what you get out of it is, what would that be? So this is DNA. Hopefully I'm not complicating the matter here. Now we have our RNA polymerases coming along, and it's going to make, this would be, what would that be? C, G, U, 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 A, right? So this is our mRNA. And as it keeps going, this obviously gets longer, right? Because it's being transcribed. So we've got C, G, U, 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 A, C, G, A. Right. Now, if these A's weren't here, this would just go like that, right? It would, RNA polymerase would just keep going. And this hairpin loop would form, but it wouldn't form within the RNA polymerase. But that's not the case, because once it synthesizes all of these A's, essentially, have I left myself enough space? Maybe not. Hang on, that's a U, sorry. Right, now our RNA polymerase, which is basically kind of about this sort of size. Right, so this is RNA pol. That huge space, this is all RNA polymerase. Right, so the DNA is inside the RNA polymerase, as is the mRNA transcript. Does that all make sense so far? Okay. However, there's a kind of limited space, right, within the, and I'm just going to move this down here. Right, here's the DNA. This is not, oh, hello there, Kim. You have to remember that this is a sequence of events, right? These are not separate, different mRNA pieces that are all present at the same time, right? So as the RNA polymerase goes along, the transcript gets longer, right? And now we have DNA, we have our mRNA transcript, and we have our RNA polymerase, this whole caboodle, right? And now it's going to be paused because it's paused on these uracils, right? And that's really important because if it doesn't pause, transcription doesn't stop, because it has to be paused so that this region of the mRNA, make sure my fingers aren't covering it up, the bit that contains the potential hairpin loop, this has to be within the channel of RNA polymerase. If it's over here, actually, if it's on my couch, which is kind of this way, doesn't make any difference. It has to be within the RNA polymerase to do its job. And the way that it makes sure that, well, not it, the way which natural selection, I guess, made sure that this hairpin loop forms within the RNA polymerase are these uracils. And again, I truly don't know why RNA polymerase stutters on uracils, why it pauses, but it does. Maybe something to do with why, like DNA polymerase stutters on triplets, right? Trinucleotide repeats. Anyway,
Dr. Crook,
go on.
it pauses basically because it's taking the time to do that hairpin.
No, it pauses because this stretch of uracils. And so typically, and this is a big generalization, polymerases don't like strings of repeat nucleotides. I don't know why. It's almost like they get confused. Obviously, they don't have a brain to get confused with, right? They're just molecular machines. But for some reason, they kind of just go, kind of like pausing here, thinking that they've already done this, but they haven't. I don't know exactly. That's kind of anthropomorphizing it, right? But for whatever reason, when you have a stretch of the same nucleotides, in particular uracils, it seems, because that's very common in bacteria, RNA polymerase pauses. And that allows these two repeats to form a hairpin loop. I'm just going to take a picture of this so I can get this off. I've kind of run out of space. Oh, shit bits. My camera's in the garage. All right, give me a second. Okay, so essentially, oh, damn it. Now my eraser's gone walkies. Okay, well, just have to use my snot rag. As you can see, oh, this is why I don't use blue on this board. I remember. Okay, so kind of going back to the end of where we're at, right? You have your DNA, you have your RNA polymerase, and essentially what I'm drawing here, this is a kind of gross simplification. This is the channel in which the DNA and the mRNA kind of coexist temporarily. And so what happens is that we have this series of U's, and oh, what was the secret? GCA, I believe.
It was from DNA. It was GCA, AAA, TGC.
And what was it in RNA speak?
It was the CGU,
Okay.
UUU. A, C, G, and then 4 U's.
So essentially what happens is this. Right? You get hydrogen bonds forming between, hang on, I didn't quite get that. That's a G. There you go. Make sure I get it right. Because these are now able to base pair, right? These form hydrogen bonds between them, as you get between opposite bases in DNA and opposite bases in RNA, and you get this hairpin loop structure form, right? And now you can already see the issue, right? We have a limited amount of space for the DNA and the RNA within the RNA polymerase. But that hairpin loop wants to form because it's very, I guess, energetically favorable for it to do so. But there's not enough space for it to fit in the channel. And so because RNA polymerase is actually a collection of proteins, it's not a single monolithic protein, it's a hollow enzyme. Essentially what that does is it levers part of the RNA polymerase away from the rest of it, and this whole complex dissociates. It just falls to pieces. And essentially that then leaves the DNA to go free. The RNA, presumably this doesn't have a great deal of affinity for what's left, and the RNA will snap back together, and then the DNA will snap back together to form the double helix, and this RNA dissociates as well. And so it's about this physical incompatibility, right, between the hairpin loop that's formed and the actual space in which it can form. And as you can see, if these U's didn't exist in the sequence, right, if this was just, you know, GCUAC, whatever, right, other RNA nucleotides, then this hairpin loop would form over here, right, or essentially the RNA polymerase would be over in that direction. Come on, love, right? And this hairpin loop would form outside the RNA polymerase, which they do, and it wouldn't do the slightest bit of difference to the RNA polymerase. So the RNA polymerase has to be paused over this potential hairpin loop to allow it to form within the RNA polymerase. So that pausing is a very, very important part of termination of transcription. That help?
Yes, a lot.
Cool.
Thank you.
No worries. All right, so I wonder where on earth my board eraser's gone. Magical mystery and board eraser. Let's get back to doobie-doo. Okay, get the chat back up. Okay, so that's one mechanism, right? The other is row dependent termination. And so this is kind of similar kind of principles, right? We have what's called a terminator sequence, and you'll see this again when we talk about the tryptophan operon in bacteria, which will be shortly after our next exam. And so essentially there are kind of two key points here. One is a terminator, right, which is another loop, right, another hairpin loop that will form. And a binding site for this protein, right? So these are features that are all encoded in the gene itself. Both the terminator sequence, the hairpin loop that will form, and the binding site for Rho. Also, another factor which is important is that there's no crazy structures that will form an RNA between those two things. Right, so essentially Rho needs a straight shot at RNA polymerase once it's bound to the transcript. If there are any secondary structures in between, that will stop it, right? So it's not going to be able to do its job. So that's another kind of key part. So kind of same kind of deal. We've got RNA polymerase kind of juntering along here, mind its own business. And it transcribes a terminate sequence. Again, it sounds kind of a little bit more exciting than it really is. It's another hairpin loop, but this one isn't in the right situation to disrupt RNA polymerase itself, but it pauses it. I don't know how, actually, to be honest. A lot of this stuff is pretty complex. And in the process, essentially what it does is it allows Rho to catch up. And so Rho is an RNA binding protein, and it kind of fancy ways, it processes along RNA in a 5 to 3' direction. So it's able to recognize this binding sequence, right, because of particular affinity for that sequence of mRNA, which is good because you don't want it binding like anywhere else, right? It has to bind specifically at the end of a transcript. And once it's bound, it will just chase. Literally, it's like a greyhound after a rabbit, right? It will chase along the mRNA and try and catch up with the RNA polymerase. And normally RNA polymerase is fairly quick, right? It will kind of continue along quite happily on its own. And so it has to be slowed down. And it's slowed down by the formation of this terminate sequences, hairpin loop. Does that make sense? And when it's slowed or paused, that allows Rho to catch up. And essentially what Rho is a type of a helicase. And so it is a helicase for DNA RNA hybrids. Is that the puppies eating the food, love? Okay. Little pests. Discovered this morning they ate my wife's headphones, which I spent about an hour fixing yesterday. I'm very, very annoyed about that. Put her out here then, love. Sorry, talking to my daughter. Okay, so essentially you have your kind of your, I guess this is DNA, right? DNA RNA hybrid, where you have your strand of DNA and you have your RNA being transcribed. And for a short stretch, that RNA is actually base paired with DNA, right? So it forms a hybrid, two molecules essentially that kind of come together. And what Rho does is it just pushes that apart and it kind of just unwinds that hybrid. And so RNA is now no longer in base pair or in contact with DNA. And there's nothing there for RNA polymerase to continue. So RNA polymerase dissociates and then it's done. So this one's a little bit kind of easier to comprehend. It's because of the direct action of Rho to force apart the DNA RNA hybrid. But again, like the main principle is of RNA polymerase slowing down or pausing, right? Both, that's important in both types of termination of transcription. It's just the thing that actually finally kicks off RNA polymerase is different between the two. And so in eukaryotes, it's a little bit different, but again, there are similarities too, right? Now, I've got Professor Luna helping me with, oh, she's just escaped the puppies, which are just busy. They're quite small, so they're trying to bite her neck and stuff. Anyway, and so in eukaryotes, there's nothing to stop RNA polymerase stopping, right? Inherently in and of itself. And so RNA transcription in eukaryotes is actually quite a bit different from prokaryotes in many ways, because there's a lot of extra steps after the pre-mRNA is produced. And one of those steps is that you have to add, so here's the pre-mRNA here. Right? It's called pre-mRNA because it isn't actually ready yet. We'll get to that in a little bit. And so it actually has to be cut off of the kind of existing transcript, and then this stuff will have things done to it, right, to process it, to make it ready for export to the cytoplasm. And so this cleavage, right, which is done by an endonuclease called RAT1, this cleavage, again, it's site dependent, right? So obviously RAT1 doesn't just go around chopping up mRNAs willy-nilly, right? It has to be very specific. So this cleavage serves two purposes. One, it frees the pre-mRNA, right? So that it's able to be finished processing. And two, now it provides a free end, right, for a nuclease to come along and chew it up, right? As you'll see in a little bit when we start on it, in eukaryotes, mRNAs need protecting. Actually, a big part of processing of mRNAs is to protect the ends of the transcript. Because if those ends are free and available, they will get chewed up by RNases. So just worth putting up some kind of terminology here. Obviously, you have DNases too, right? Does the same thing. That's what we were putting up DNA in the fridge for, so that they wouldn't be active. Can anybody remember an example of an exonuclease that we've come across already? Or exonuclease function? Cuts off the base of the end of a strand, newly synthesized in this case. In case that base is wrong. Telomerase, good try. Not quite. That's actually a type of, it's a real funky ass kind of RNA DNA polymerase type thing. I'm not even sure what telomerase is, to be honest. I kind of know what it is, but not well enough to explain it. Something else involved in DNA replication. What stops, goes backwards and cuts off the wrong DNA nucleotide and then continues? It's DNA polymerase, right? So DNA polymerase has exonuclease activity. And it's very specific. It's 3' to 5' exonuclease activity, right? So it cuts off the 3' most base if it's wrong. So RAT1 Actually, I'm not. I'll have to read into this more. It's always been a little confusing whether rat1 actually creates this cut or if it just degrades the mRNA once it has been cut. I'll have to double check that. Where's my pen? Make a note. It might do both, but it'd be nice to be clear on that. Anyway, once, so yeah, RNA processing, we'll get to that once we finish this. But this free 5' end is now like a nice juicy target for an RNase. And so rat1 binds to that, and it specifically digests the mRNA in a 5' to 3' direction. And so essentially, rat1 is going to start digesting this leftover tail, essentially, while RNA polymerase is synthesizing more of it. Okay, does that idea make sense? So essentially, there's going to be this race between RNA polymerase to make mRNA or make more RNA, and rat1 to chew it up and catch up with the RNA polymerase.
So rat1 is not actually protecting the ends of the mRNA.
It is most definitely not. It is chewing them up. It's like a Pac-Man.
Okay, so it's like catching up with the production over here, and which one wins gets the if
prize.
one eats more or the other one produces more, right?
Correct. Yes. Okay. That's exactly the idea. Actually, I wonder if anyone has. I always remember this Stephen King novel called The Langoliers. I don't know if anyone's ever read that, about these things which go around destroying the world of the past. Like when a new day starts, the old world is anyway. It's kind of neat. You should look it up. It's a cool story. Anyway, so essentially now we have a race that's on between rat1 and RNA polymerase, and rat1 is faster. And so essentially what happens is that even though RNA polymerase is still moving along, it's still adding nucleotides to this mRNA, I guess, not even sure what to call it, to be honest, stub of RNA. Rat1 catches up with it and destroys that. And this is actually kind of a neat idea if you think about it, because these RNA bases they cost, right? They're not free. They cost energy to produce. And so this bit of RNA is complete waste, because the actual pre-mRNA, which is going to be used for translation, has already been cut off, and that's busy being processed and exported. And so this little stretch is completely useless. And so essentially what rat1 does is it recycles that mRNA, or those RNA nucleotides. Those will then get kind of refreshed, I guess, and used again. And once it catches up with RNA polymerase, it again physically punts RNA polymerase off of the DNA. I'm not entirely sure how that works specifically. And I'm not sure if that's just because I don't know it, or if it's because it is not known how that works. Again, I'll have a little read into that, see if I have some kind of answers for you by next class. And that's it. And so, and another thing that I really, really want to get across too is that while this RNA polymerase is being booted off, whether it's in bacteria or in eukaryotes, it doesn't matter. There are other RNA polymerases on that stretch of DNA transcribing that gene too. So, this is a good picture to use, right? So as soon as this RNA polymerase has moved off of the promoter, right, it's physically no longer blocking the promoter, this promoter is now free for another RNA polymerase to come in and bind and start transcribing the gene. So it is not the case that a gene is transcribed once, finished, and then it's transcribed again. Depending on the strength of that promoter, you can have multiple RNA polymerases transcribing the gene at any one time. And if you think about it, that's a pretty good way of, if you need something very quickly, really kind of amping up production, right? To have multiple machines basically doing the same thing at the same time. It's like having like 4 or 5 Roombas, right? You know, if you want really clean floors. That's a bad analogy. Best I could do at short notice. Okay, so again, while this is going on over here, you know, upstream, there'll almost certainly be another RNA polymerase producing another copy of that same transcript. And that's actually particularly important in prokaryotes. Any questions about any of this jazz? No. Okay. Cool. Okay, so let's move on to Lars. So essentially what we've done is so far this bit, right? And you know that this picture here is a picture of a eukaryote. How do you know that? Huh. I know it. How do you know it? What's the key defining feature of eukaryotes versus prokaryotes?
Isn't it the nucleus? It's not enclosed.
In which?
The prokaryotes, it's all mixed up with the DNA.
Right. So prokaryotes don't actually have any organelles, right? So there is no internal partitions. The DNA tends to kind of clump in one particular part of the bacterial cell, right? It's not always uniformly spread out. Kind of associates with more of one area. But yeah, there are no organelles, right? So this nuclear membrane. Oh, that's a good one, Ali. Yeah, prokaryote, no organelles. That's neat. I remember that. So we've got essentially a couple of things that have to happen before translation can occur in eukaryotes. Now, again, remember in prokaryotes, this is all happening in the same space at the same time. So in prokaryotes, as soon as that mRNA is produced, or enough of it, that a ribosome can recognize it, it will start getting translated at the same time as it's being transcribed. So prokaryotes have, not only are their genes collinear, right, between transcript and protein, but they're also transcription and translation occur at the same time in the same place in prokaryotes. That's actually critical to how prokaryotes, bacteria, control the expression of some of their genes, which again we'll get to later. Now in eukaryotes, that's different, because in eukaryotes we've got our DNA all packaged nice and neatly in our nucleus, and our translation occurs in the cytoplasm or the rough endoplasmic reticulum, depending on where those proteins are destined to go in the cell or outside of the cell even. So we need to do a couple of things. We need to process those mRNA transcripts, or process them into mRNAs. And we also need to get those mRNAs, those mature mRNAs. It's not like they're, oh, I'm so old and wise. It's just that they've had all that stuff done to them, into the cytoplasm, where they will then be available for ribosomes to assemble and translate them, right? So now we need to do this kind of bit in the middle, essentially. And there's 3 different kind of parts of RNA processing. 2 of them are all involved, well, largely involved with protecting the ends, right? So you have 2 ends. You've got a 5' end of the transcript. You've got a 3' end of the transcript, right? So you need to kind of protect those ends so they don't get degraded. What else do you need to do? What needs removing from the pre-mRNA in eukaryotic genes? What things are there which cannot be there when translation begins? Think back to eukaryotic gene structure. I love asking questions because it gives me time to rest my voice. I feel like I should be like those Hollywood voice actors. You know, I get like extra pay from my union for, you know, using my voice. What are the features of a eukaryotic gene? Think of some names. I'll give you two. We've got a 5' and a 3' UTR, which stands for untranslated region. What lies between those two? 5', 3' UTR. In between, we have two different things, one of which codes for protein, one of which does not. Anybody remember the name of the thing that codes for protein? Most likely, you know, unless you kind of married your childhood sweetheart, you have some of these. That's a little bit of an off-color joke, sorry. X's, exons, right? So exons code for protein. What is between exons that does not code for protein? Yay! Thank you, Ali. Introns. Okay, so we've got to get rid of those introns, right? Because if we don't, they will be translated. And if they're translated, that is usually catastrophic because they will contain essentially gibberish and almost certainly will contain stop codons, right? So you'll get a truncated gibberish protein, which is just never good. Okay, so those are the two kind of general things. You're going to cap the ends, the 5' end and the 3' end, and we're going to cut out the introns, or we're going to splice them out, right? Is the kind of fancy word for that. And so what we want to end up with down at the end, this is not my art, but it does the purpose, right? Is we have a 5' cap. We'll talk about that in a bit. We have a poly A tail, and that serves other purposes, right? We have a 5' and a 3' UTR, again, untranslated region. I'll type that up for you. Oh, if I could spell region properly. And just because they're not translated doesn't mean they're not important, right? These UTRs are very, very important for a bunch of different reasons. Transcript stability, ribosome binding, all kinds of jazz. And in the middle, we have our open read-in frame or coding sequence. That's what ORF stands for. CDS is pretty much the same. There you go. And so this bit here, when we have our start codon here, which is an AUG, we have stop codons here, which are one of these three. This is what contains the information that is going to be translated into a protein. And these various steps occur as the mRNA or the pre-mRNA is being made. Go on, Kim, I'll let you have a drink. I think someone was making their lunch the other day while I was giving a talk that obviously shouldn't point that out, I guess. But anyway, so as soon as the 5' end of the pre-mRNA gets made, it gets capped. As soon as an intron emerges, it gets spliced, right? So this is a process that kind of happens as RNA polymerase is moving along. Any of the mRNA coming out is going to be processed as soon as it becomes available, right? So this isn't something that waits, again, for a fully produced pre-mRNA to be made, because if it is, that 5' end has been flapping around in the breeze for a while and it'll be degraded, right? So it has to be capped as soon as it emerges from the RNA polymerase. Ooh. So the very first thing to do is to put on that cap. And this is called a 7-methylguanosine cap. Also shortened to, oh God, yeah, Kim, I'm totally missing my coffee. It's always funny because this is actually my prime time regular scheduled coffee drinking spot, like right now. And so if I have coffee before I start my class, it's too early. And then if I have it afterwards, it's too late. So yeah, it's all your fault, you lot. Blame me for it. I should have should have some coffee sweets or something kind of on the side. So this 5' cap has actually two purposes, right? It's both involved in protecting the 5' end from exonucleases, from RNases that will come along and chew it up. And it's also, in eukaryotes at least, what is recognized by the ribosome, or at least the small subunit of the ribosome. And so this is really important because this is not just to protect the RNA, it's actually a key part of translation as well. And this is basically what it looks like, right? And so these are all kinds of funkiness, right? In terms of, these are kind of sort of based on nucleotides-ish, right? But they have all of these methyl groups kind of dotted around them. And so essentially, and this down, well, actually, crap, I'm not even sure which end it would be attached at. Right, down here somewhere, that's it. Down here is the actual start of the mRNA, right, kind of down from this part. And so these are not recognized by any RNases because they don't look like RNA. It's like RNAs are like, what the hell is this shit? And it'll go off and degrade some other RNA somewhere else, right? So RNases would not recognize this. And so it is then, as soon as this thing is added, that end of the pre-mRNA is protected. So it's like the pen lid, essentially, put on the cap of a pen. And this is you'll often see it referred to as a 5 mpg cap. Okay. All right, now we've got a fairly kind of chunky bit to talk about here because this is splicing. This is fairly involved, to be honest. Yeah, it's kind of like one of the worst things to leave to the end of a lecture, unfortunately, particularly when it's an hour and 15 minutes long. So if you have any questions, please ask them. It'd be nice. So essentially, these introns have to be removed. And so this is done by a whole bunch of different players. And so the very first part begins with the actual sequence of the mRNA or the pre-mRNA. Right? And so this is essentially showing the first intron in a pre-mRNA. So this is obviously the first one to be produced or emerge from the RNA polymerase. These vary in size from, 50 bases to, I don't know, like 10,000 bases long, right? So there's no uniformity in their size. And really, apart from a few bits, there is no uniformity in their sequence, right? These do not code for anything, at least that we're aware of, right? There is potentially other stuff in there. And so there's no particular strong natural selection or selective pressure against mutations that occur in the introns. Right, so these are hugely variable, nothing very much to compare them in both size or sequence, but they do have specific characteristics. And the key ones are these two bases here, these three bases here, and this base, right? So when you do a comparison between, introns, actually, when I was doing my PhD, I actually used to look for these things. Isn't that crazy? By eye in sequence. This was before I actually had a computer, but it didn't do this. And what you're looking for in DNA is a GTAG sequence, or in mRNA, it would be GUAG, or in this case, CAG is a little bit more precise. What these are called are the splice acceptor points. These are the bases that are recognized by the spliceosome, the machinery that cuts this intron out. And so these bases, these two specifically, and these three are absolutely critically required for splicing to occur. They will be present at the beginning and at the end of every intron. And actually, there are a class of mutations called splice acceptor mutations. And so if one of these nucleotides gets mutated in DNA, so now it's instead of GU, it's GC, that intron will not be spliced out. And so that will remain in the mature mRNA, and translation will occur into the intron. Right, just shows how important these bases are, these GU and the CAG. If they get changed in any way, that intron will not be removed. Right, so again, kind of not entirely analogous, but similar to kind of the, like the inverted repeats and transposons, right? These are what are going to be recognized by the machinery that will remove them. And then the other conserved kind of sequence feature is this adenine here. This is going to be used for the actual removal, right? It's kind of like funky in the sense how it's done. But this is, oh, actually, hang on. Questions to the class. What's one of the main differences between DNA and RNA nucleotides? There are several. Give me a couple of differences. Uracil, good. Single strand, mostly, not always, as we've seen, right? Location, maybe. What else? Actually talking about the sugar part of the nucleotide. Opposite of that. What does the D in DNA stand for? Deoxy, right? So in RNA, it's not. So in RNA, you have two hydroxyl groups. Right, they have one on the 3' OH, which is the same as DNA. They also have one on the 2' OH, and that's going to become really important with this adenine here, as you'll see in a moment. Okay, so kind of keep in mind, we have our exon 1, we have an exon 2, and within our intron, we have these very important conserved sequences. Essentially, if something's very highly conserved, that means it is very, very, very important. Because if you have a mutation that changes this, that gene no longer works. Right? And so if that gene is involved in something important, that organism no longer works, right? So there's a very strong selective pressure against any changes to these sequences. It's kind of another way of thinking about it in kind of evolutionary terms, right? The reason why these are conserved sequences is because they are so important that if you screw them up, you screw up an awful lot of important stuff. It's kind of like another way of looking at it. I'm talking to the whole biologist here, right? I hope you like my holistic approach to biology education. Anyway, so these are the important points. And now we're going to see how those work. Is there anything else in here that I need? Oh, yeah. SNRPs. They're like Smurfs, but they're smaller and made up of RNA and protein. Right. So I love the word SNRP. Right. I'm not going to type it out because, boy, I don't have enough time left in the class. It's actually written up here for you. That stands for small nuclear ribonucleoprotein particles. See why I call them SNRPs. And so if you cast your mind back, RNA polymerase 2 in eukaryotes makes mRNAs and small nuclear RNAs. All right, so small nuclear RNAs actually are these. Actually, I never know what happened to U3. Perhaps, you know, it was kicked out of the band or something. Oh, that's a bad joke. Sorry, you can see my lack of caffeine is affecting my humor. I have to make a note of, like, write down some of the dad jokes that my kids tell me, so I can share them with you all. Anyway, so these 5 small nuclear RNAs, U1, 2, 4, 5, and 6, are really, really important. They're actually the catalytic parts of this process, right? And they complex with a bunch of other proteins to make SNRPs. And those SNRPs are what constitute what's called the spliceosome. Right, the spliceosome is essentially a conglomeration of these things, which together, it's all right, dog, I'm not bothering you, which together cut out the intron and stick the exons back together, right? This is the key. You know, you can't just cut the introns out because then you'd have just fragments of mRNA, which would be no good to anybody. You have to actually stick it back together again. Okay, so this is the general overview of what happens. I'll show you in detail in a little bit. So we have our 5' and our 3' splice acceptor sites. That's the 5' of the intron and 3' of the intron. And essentially what happens is that the mRNA is cut, and that cut end is bent back and it's actually stuck to that A. So remember we had that conserved adenine, the branch point, which is a short distance upstream of the 3' end of the intron, right? That's important because that basically serves as like a docking point for the 5' end of the intron as it's bent round and stuck back onto itself. And so here, the reason why I was banging on about the 2' OH group, that's actually now used to stick the 5' end of that intron to itself. See, this wouldn't work in DNA, right? Because there's no OH group there. Obviously, you'd hope it wouldn't work in DNA because that would be terrible, right? So now we have what's called a lariat loop, which is always fun to talk about in Texas because, you know, cowboys and stuff. And the 3' end is now cut off, right? So now we have essentially 3 bits of mRNA. We have our exon 1 and the 5' UTR. We have our lariat loop, and we have the rest of the pre-mRNA, exon 2 and onwards. There's typically like 3 or 4 exons at least in a gene. And so now we need to stick that end of exon 1 to the end of exon 2 to now get our continuous mRNA molecule. Does that make sense? Because it's going to get crazier. Obviously, how this works is non-trivial. And so it all involves the schnurps. It's like you pronounce it like schnapps, like German liqueur, but schnurps. That's how I'm saying you should pronounce it. I don't actually know how people pronounce it, but that's how I do it. Anyway, so essentially what you have first is recognition of the 5' acceptor site there by U1, recognition of the branch point by U2, where the streets have no name. Probably most people wouldn't get that. Never mind. I was a big U2 fan when I was growing up as a teenager, many, many moons ago. And so then we have another whole bunch of stuff. And basically, its job is to bend this intron round and hold on to the exon 1 once it's been cut off. Because you don't want this exon 1 just floating off, right? And so as this lariat loop is being formed, it can only form by having cleaved the pre-mRNA here at this 5' acceptor point. And so the spliceosome keeps hold of exon 1 and the 5' UTR as it's bending the intron round to stick it back to itself. And then U5 both cleaves the 3' site here and religates exon 1 to that now free end of exon 2. And in doing so, all of that jazz gets released with the lariat loop, and we now have a continuous mRNA species. So again, initial recognition of the 5' splice site and the branch point. The rest of the spliceosome comes in, cuts the intron at the 5' end, holds on to exon 1, bends everything round and sticks the end of the intron back to itself. And then the next key part is cutting of the 3' site and sticking exon 1 to that now freshly cut exon 2. And that generates two things. One, the spliceosome with the lariat loop, and the other is a processed, at least for that intron, piece of mRNA. And then these disassemble and this RNA gets degraded back into RNA nucleotides to be recycled and used again. Anybody having questions? No? Okay, so again, oops, I don't know why I have that up, my calculator. Again, important parts of the splice sites and the branch point. Key players are the small nuclear RNAs and proteins, which together are called SNRPs. These form the spliceosome, which is what does the job. And then it cuts out the intron and sticks the two exons together. Now there are typically multiple introns. Sometimes you only have one, can happen. It's somewhat rare. But this will happen whenever the spliceosome assembles around an intron. It doesn't have to be in order. First intron and then second intron and third intron. It's just whatever. It doesn't really matter. As long as they all get cut out, then you're good. Okay, so kind of looking at this in like a linear process, right? So the very first thing that's done is the capping of the 5' end, the 5-MPG, 5' MPG. Splicing machinery can assemble as the mRNA is being transcribed, or if it's busy, it doesn't matter. It will just catch up at a later point. Oh, someone's making toast. And that will simply keep happening until all of those introns are removed. Right, again, doesn't necessarily happen in the order in which those introns are transcribed. Also, there are some proteins, and this is something I don't know a whole bunch about, which are recruited to these splice junctions. Even though the mRNA is intact, that serves as like a binding site for proteins that are involved in RNA export from the nucleus. And then the final part is something called polyadenylation, right? And so this is to produce a long series of A's, a poly A tail, right? And you're probably wondering, like, why would we want that, right? And so poly A tail is essentially a buffer at the 3' end of an mRNA. And it's also a site for poly A binding proteins. So the poly A and those binding proteins, those poly A binding proteins, those then protect the 3' end of the mRNA. And I think I'll go into that in a little bit more detail next class, because we've got slack in the schedule. All right, cool beans, everybody. So one thing as well that I'm kind of trying to do as we go along as well, because I want to help prepare you for when we get to the complexities of a control of gene expression, is a lot of the features that you're seeing now are very important in the control of gene expression, right? There are lots and lots of different ways in which you can control gene expression. And one of those ways is by controlling stability of the mRNA transcript. So this is not a static process, right? This can be modulated to control how long this mRNA hangs around for. Because as long as that mRNA is in the cytoplasm, it will be translated by proteins. And so kind of, you know, I've been talking a lot about stabilizing, right? Protecting the mRNA transcript. Well, you also don't want to protect it forever if you don't want it to be translated forever as well. So that's kind of the flip side, the yin yang of this whole deal. Anyway, that's it for now. I've hit my limit. It's time for make a cup of coffee. So I shall either see you tomorrow in lab or I shall see you on Thursday. So have a great rest of your day, and I'll see you soon. All right.
Mr. Cook, I had a question really quickly.
Oh yeah, whew, that was quick. Go for it.
I had a question about the lab actually that was due last week, the second one.
Oh, which one was that? The pipetting one?
The graphs one where you had to do the
Oh.
graphs. So
Yep.
I wasn't in lab on that week before because me and my little girl were sick with fevers. So I know I completely bombed that lab because I had no idea what I was doing.
Okay.
I like tried my best on the graphs, but I have a feeling I bombed it and I probably didn't even get one thing right on there. So, and I was going to ask you that Thursday for help, and that's when you had canceled that class on that Thursday.
Oh, right. Well, I mean,
so I don't know if there's a way that like I can redo it, or is there a way that you can explain it or anything like that tomorrow?
I mean, without a doubt. Are you in lab in person tomorrow?
Yes.
Okay, so tomorrow should be a little bit of a short lab, relatively speaking, because I think, I don't actually know yet, I'm waiting to hear back from the lab technician. I think we won't be doing at least 2 of the 3 things that we need to do or would have done normally. And because I've opened up all labs, all of them, right? Not just ones going forward, but all of them from the beginning of the semester, to be due by just after the end of classes. You should be able to go back and change that. If not, I can quite easily just cancel that for you, and then you can redo it. It's not really difficult.
Okay, because I know you had kind of touched base that Thursday after I had missed class, and then I completely I didn't understand it, and I was still really confused, and I was like, oh, I asked him on Thursday, and then I woke up that morning and I was like, oh, class is canceled.
Yeah. Well, technically not canceled because there was a YouTube video that you need to watch, but yeah, I wasn't available, unfortunately.
But the in-person for the where I could ask questions was wasn't there, and I was like, oh man.
Yeah, no worries. Hang back after the end of lab tomorrow, and I'll go through it with you.
Okay, cool. Thank you so much.
No worries. Cool. All right. I'll see you tomorrow.
See you tomorrow.